Why did the football coach go to the bank?
Get a quarterback.
He's a quarterback. Sit down. What asks no questions but has to be answered? What asks no questions but has to be answered? Doorbell. Okay, how about this one? What type of music do mummies sing in the shower? Rap music. Got it. This is where we left off, and this is where we're going to pick it up. We talked about three main types of filaments. We have the thick, we have the thin, and we have the elastic. And we're going to get into some more details, bless you. We're going to get some more details about each one of these. We're going to dive deeper into the microarchitecture. Okay? So this picture should look familiar. And this lecture and the lecture on Monday is where we're going to get into a lot of the mechanism. So it's important for you to come to SI and ask those questions of, I don't get it. And what aren't you getting about it? What are you not understanding? Because we're going to talk about a lot of moving parts today and Monday. The thick filament is this purple structure, and if we blow this up at higher view, higher magnification view, you can see it in the upper right. And the appearance is a much thicker filamentous protein than its counterpart, which is blown up down here, which is not to scale, right? Because these look about the same thickness. But if you look here where this drawing is to scale, you can appreciate that this reddish-orange filamentous protein is blown up as shown here, and it is a fraction of the thickness of the thick filament. Okay? That thick filament is mostly composed of a protein known as myosin. And for our purposes, we're going to say that it is only myosin. And that myosin protein has a head associated with it. It kind of looks like a golf club. It's got like a long shaft, right? The Masters starts tomorrow, for those of you that are paying attention to it. Tiger's actually playing, for those of you that care. Okay? At least he's going to start. We'll see what happens. It looks like a golf club where there's a shaft, and then here is like the head of the driver. Okay? But this protein is actually two-headed, so it'd be kind of like a golf club with two heads sitting on it. Some of you are like, well, that's what I need to improve my game. Okay? I could totally not miss it if I had two heads. Okay? Mine would probably just shank harder to the right. Okay? That's the architecture of this protein, this filament. If we look down here at the thin filament, again, keep in mind that its relative thickness is shown here, and you blow it up, we've got more than just one component. We have multiple components. So I want you to talk to your neighbor and see if you can explain the different types of proteins that are shown here, and what are they responsible for. Okay? Ready, set, go. What did you come up with? So who can give us an explanation of the importance of tropomyosin? Anybody? So many of you are volunteering, I just don't know what to do. Okay, here we go. Tropomyosin moves. Explain. Thank you. In more detail. Okay, so it moves. Tropomyosin definitely moves, and it sort of unwinds and kind of, it gets out of the way, because it's actually blocking something. Okay? it does bind to something, but it doesn't bind to myosin. Okay? Close.
So the G-actins are what make up the strand. The tropomyosin blocks the calcium binding, so that prevents the muscle from firing when you don't want it to. And I think the troponin complex binds that to either the myosin filament or the
sarcosome. Close. So the three of you are circling this whole concept. Okay. Anybody else want to try adding in some detail?
Doesn't calcium bind to troponin, causing tropomyosin to shift, and then that's like where the movement happens from myosin?
That was beautifully done. Do you want to take this? That was. Did you guys hear that? No. Step number 1. Calcium binds to the troponin complex. The troponin complex has 3 specific proteins in it. In this class, we just say that it's a complex. We're not going to discriminate amongst the different subunits. But calcium binds to troponin, that yellowish, blobby-looking protein. Step number 1 that was said. That's totally accurate. Then there's a conformational change. And this, I'm going to elaborate a little bit. This tugs on tropomyosin as this complex changes shape because troponin and tropomyosin are physically connected. So when one shifts shape, it tugs on the other one. And as was set up earlier, tropomyosin then actually kind of slides out of the way. Out of the way of what? Well, there is a binding site that it's covering. The binding site is where these myosin Gulf head looking structures are going to bind. And that binding site is located on the G-actin protein, which are these red balls or red spheres. It's a globular protein that makes up the bulk of the thin filament, but it is incorrect to say that the thin filament is made up of actin. Did you follow that? The thin filament is made up of actin and tropomyosin and troponin. Now, if you didn't catch all of that flow, don't worry, we're going to go over it numerous times with lots of different pictures. But that's the moving part that we need to understand, okay? Then the last filament is this elastic filament that we said was anchored at the Z-disc. We said it's a Z-disc because it's kind of zigzag. And after contraction is done and relaxation is going to take place and you're going to elongate the muscle, the elastic filament known as titin helps with that. It's like a rubber band that once it's done stretching. Have you ever played these games where it's like a carnival game and you get in and you have this strap and there's this giant like bungee cord attached to you and you start here and you run as hard as you can, you're racing a friend and you have to take like this bean bag with Velcro and see if you can, who can put it the furthest away before the elastic band snaps you back. Have you ever done that? That's kind of the activity that titin has, is it helps with this elastic recoil so that it comes back to its starting position. Okay? So these are the main players that we're going to be talking about today. If we take a peek and characterize them in these terminologies, we can talk about contraction and regulation. If it's contraction, then it is fully responsible, or it fully contributes to actin and myosin seeing each other and allowing for that myosin head to bind to the actin molecule. And then later what we're going to see is, remember when I talked about this and we see how these are brought together? It's sort of a ratcheting effect that takes place. So it binds, it releases, it binds and it releases, and it ratchets this direction as they contract. And then when they relax, it lets go and cocks back, and this actually snaps back to its resting place. So the proteins that are involved in active contraction are considered contractile proteins, and that would be myosin and actin. It would not be correct to say the thick and thin filament. Why? Because the thin filament has troponin and tropomyosin as well as actin. Okay? Now, regulatory proteins. These guys influence or modulate or regulate whether or not actin and myosin see each other. So we call them regulatory proteins. And the way that they regulate it, as we just said, is there's a lot of white space here if you want to write some of this additional detail down that we talked about earlier. Calcium binds to the troponin molecule. This is the same picture that we saw before. This is just a larger view. Calcium binds to troponin, causes a conformational change. That tugs on tropomyosin, and it shifts out of the way and exposes a binding site behind it. So these guys, in the presence of calcium, or in the lack of calcium environment, they'll either block or reveal the binding site so that these two guys can see each other. Now, if calcium is so important, how do we regulate calcium? So we're back to this picture that should look somewhat familiar. We used this picture last time. We spent a lot of time. We didn't get very far slide-wise. We covered a lot of material because there's a lot of explanation of this figure. If you're unclear on this figure, that would be really valuable to go over an SI. See if you can draw it. Maybe just draw components of it. What's easier to draw is that figure down there, because that's a two-dimensional view of this figure, and specifically what component. Right up here. You see up here? So if you're not artistically gifted, like myself, you may resort to a 2D picture, and that's fine. We're back to our triad. The triad we defined earlier, last lecture, but we didn't really call it a triad because I didn't want to throw too much info at you. We defined the components of the triad. Triad means three, right? So in the middle is our transverse tubule, abbreviated T-tubule. The T-tubule is what? Who can define what a T-tubule is? We talked about it last lecture.
That's the extension of the sarcolemma into the interior of the myofibrils?
It is. It's the extension of the sarcolemma, which is the membrane covering of this particular cell, that allows that electrical signal coming down the membrane to dive deep into the cell and be able to send a signal into the cell where the middle component of the myofibrils can see it. Now on either side, you have a triad, so there's three. So down the middle is the T-tubule. On either side is called the terminal cisternae. Who can define what that was as we defined it last lecture? Terminal cisternae. Uncomfortable pause. What does the word mean? Can you guys define the word? Terminal? End. Right, you go to the airport terminal, right? That's as far as you can walk, because then you're flying. Terminal is like the end. And cisternae, it's the Latin word cistern for, it's a component of the sarcoplasmic reticulum, but the word cisternae means what in Latin? I'm going to take a drink out of the cistern. It's actually a tank. It's a little bit less elegant than a cup. Okay? Okay? So cistern is a tank, like a giant storehouse, if you will. Someone shouted out sarcoplasmic reticulum. That's correct. SR. The SR up here, the sarcoplasmic reticulum, is this blue network that is a storage facility inside the cell for calcium. The edges of the sarcoplasmic reticulum, also shown in this blue, but in larger diameter, is the terminal cisternae, the end tank of the sarcoplasmic reticulum. Make sense? So given the elaborate network of blue, SR, and larger blue tubes, terminal cisternae of the SR, do you think there's a lot or very little calcium stores inside this particular cell? A lot. A ton. A bunch. Okay? To use very scientific terms, a buttload. Okay? There's a lot of calcium that can become available, but look to see where it's stored. It's stored in this intricate network so that it's not freely floating inside the cytoplasm. Or another word that we would use is the sarcoplasm, because the sarcoplasm is the inside fluid environment of the muscle cell. Instead of the word cytoplasm here, we use the word sarcoplasm. Two words that mean the same thing. Okay? Rest on that topic about calcium and why we would spend so much biologic energy making this elaborate network to store calcium so that it's not just readily floating around. You're going to see here in a little bit why that is. So what determines calcium availability is whether or not it's inside the blue tubes or whether or not it's outside the blue tubes. If it's outside the blue tubes, the sarcoplasmic reticulum or the terminal cisternate of the sarcoplasmic reticulum, is it in the cell or out of the cell? It's in the cell. Where specifically in the cell is it? It's in the sarcoplasm. Okay, did you follow all of that explanation?
Can you do it again, please?
Okay, can you do it again? If you have calcium that's outside of the sarcoplasmic reticulum, or it's outside of the terminal cisternate of the sarcoplasmic reticulum, is it inside or outside of the cell? It's inside the cell. Where specifically is it inside the cell? It's within the sarcoplasm, which is the fluid environment of the muscle cell. In a general biology cell, we would call it the cytoplasm, but here we call it the sarcoplasm. Okay, good. So now we can shuttle calcium in and out of the cell. Instead of sending it in and out of the cell outside the cell, we send it in and out of the sarcoplasm, but within a storage tank located in the cell. Okay? All right. When we take a look at how this internal sliding or this interaction, this is where we kind of left off topically with the last bullet point on Monday. We have these filaments that slide across each other, create tension, force. But if they just slide across each other and they're not tugging on anything, nothing happens. Okay? Because you can't translate the force outside the cell. You have to translate the force outside the cell. So if this is our two-dimensional view of thick and thin filament interaction, and they're sliding back and forth all day long, that's wonderful. If you don't link them to the external environment outside the cell, you're never going to get translation of any force. You won't get muscle contraction. So the analogy that I used in the last moments that we were together last time is driving on ice, and your tires move on ice, and they could be rotating super fast, but if they're not actually grabbing or making frictional contact, you're not going to go anywhere. You have no translational force. You have tons of rotational force, but no translational force. So we have these accessory proteins that anchor the myofilaments, and they regulate the length of the myofilaments, and they allow it to link to the outside environment and translate that force. One of the most clinically relevant is dystrophin. Dystrophin is this reddish-colored protein right here that specifically links the thin filament, right, to other linking proteins that cross through the membrane and attach to this outside environment of the cell. So it links this outermost myofilament to the transmembrane protein, allows for this connection to take place, translational force. Now, if you have a genetic defect in any of these linking proteins, like in dystrophin, you're going to have a debilitating muscular disease, and that's known as muscular dystrophies. That's where it gets its name. So the one that I want to characterize, and there are quite a few, but we'll just introduce this one. It tends to be one of the most dangerous, if you will. But the whole group of muscular dystrophy is a hereditary disease that causes degeneration or weakening of the muscles. And over time, they're replaced with fat and scar tissue. So this example of when they slide, or when you have this spinning wheel and nothing grabs, is you're getting a lot of contraction that takes place, but you get nothing connecting it to the outside environment. So it creates a lot of frictional scarring over time. Okay, so the muscles can contract, but they can't necessarily do anything. And then they sort of just disintegrate over time as they sort of frictionally wear each other out. The one that I want to characterize in most detail for next Wednesday's exam is Duchenne muscular dystrophy. This is a sex-linked recessive trait, so it affects about 99% of the affected population are boys. There are some rare occurrences of girls that get it, but mostly it's males. Okay? Usually it's diagnosed between 2 and 10 years of age. And you'll see abnormalities within pediatric milestones of muscular development. So that's why at 2 years of age you can see it. They have trouble with walking. They should be walking by 24 months. This is the most common form of the entire category of muscular dystrophy. And this mutation is specifically in the protein dystrophin. The actin is not linked to the sarcolemma, and so when they contract or you get sliding, it causes damage and eventually cause scarring and necrosis over time. Life expectancies of these individuals are on average 25 years of age. Now that number is increasing slowly over time. Okay, but in this population that should hit home because most of you are probably less than that, but you're rather close, just a few years off. So the effects here in dystrophin are conserved with respiratory muscles as well as cardiac muscles. Respiratory muscles, in fact, are skeletal muscles. But dystrophin is found in cardiac muscle as well. So there are cardiovascular and respiratory failures that take place in these patients. So it's really incurable at this point in time. Okay. Questions?
Does it mean that the only one myofibril is linked to the cell membrane of sarcolemma, which is responsible for pulling all of the other myofibrils inside?
No. There's lots of dystrophin that's found throughout the entire network.
Okay, yeah. So dystrophin connects ultimately all the myofibrils together, and then the dystrophin on the most outer perimeter of myofibrils connects it to the sarcolemma.
The dystrophin connects to the actin, specifically to the thin filament actin. That's where it links to. Okay, so you have dystrophin connections throughout the entire myofibril to the outside environment.
Oh.
Because when you contract a muscle, you may not use all of your myofibrils. It's called recruitment. We'll talk about that in the subsequent lecture. Okay, so you'll be efficient with your energy. Right? So if you want to pick up this laser pointer, right, and you want to bring it here, there's no reason to recruit every myofibril in your biceps brachii. Right? Because it's not, there's no point in, doing it like this. Right? But if you're going to pick up, a dumbbell, you have visual cues that you're going to recruit probably a vast majority of those myofibrils. So recruitment depends on motor unit stimulation. So we'll talk about that. So you don't necessarily, it's not an all or nothing thing with their muscles. Right? I mean, when you do a high five, it could be like a nice high five, right? Or you could just like smack, right? I mean, so there's different levels of force production depending upon the number of myofibrils you recruit. All right, neuromuscular junction. If we look at our neuromuscular junction, we have this relationship that occurs between a skeletal muscle and the nerve that actually innervates it, kind of like what we were just talking about, capping. And the muscle itself isn't going to contract and do anything unless it's stimulated by a nerve. So what happens if you poison or if you sever that particular nerve? You get what we call denervation atrophy. So you'll get shrinkage of that muscle or a loss in size of the muscle just by not sending nerve impulses to it. Okay? So if you have a patient who has a spinal cord injury, you will very quickly see if their paraplegic or quadriplegic, depending upon where the injury is. Paraplegic would be not any use of the legs. Quadriplegia would be no use of all 4 limbs. But very quickly in the first 2 weeks, you're going to see major atrophy of the muscles because the nerves are not sending a basal level of electrical signal to the muscle. And then you'll see additional atrophy from a lack of use. Okay, but the initial atrophy is due to the denervation. So when we talk about this relationship, and we were just mentioning this, we need to talk about motor units. So these motor units, a motor unit is defined as a nerve fiber or a neuron in all of the muscle fibers or muscle cells that it innervates. So collectively it's considered a motor unit. So we've got somatic motor neurons that are nerve cells that serve the skeletal muscle, right? They're efferent pathways. We have somatic motor fibers that are the axons that lead to skeletal muscle. And we get this branching pattern that can take place so that a nerve fiber doesn't necessarily always have to innervate one muscle cell. Okay? So one nerve fiber could go to multiple muscle cells. So like an example that I would use is like Christmas lights. Right? So anybody have familiarity with hanging Christmas lights? Sometimes it's the most frustrating activity on the planet, right? You're supposed to be in the mood and Merry Christmas, neighbor, right? And so a lot of times you set them up so you've got like, maybe you have a timer. And you have one timer, and then you have a bunch of different light strings going to that one timer. And if everything works the way it's supposed to, as the timer goes on, and all of the lights turn on at the same time, right? And have eggnog, and you wait for Santa Claus, and everything's great. So that one control unit can serve multiple different pathways. And so that's kind of how this motor fiber can actually be organized. If we look schematically at a diagram, you can kind of appreciate the way this plays out. So for example, here's our spinal cord prep. You can see red and blue colored pathways. You've got motor neuron number 1 and motor neuron number 2. Number 2 is colored in red. If you have a black and white print, it's going to be a little bit more complicated to discriminate the differences. But you can see very clearly on this colored image that the motor unit number 1 that's blue goes to the blue muscle fibers. In motor unit number 2, the red goes to the red muscle fibers. And they're all coming out of the anterior root horn, which is the motor side, which makes sense. But depending upon the activity, you may say, okay, if I'm picking up a laser pointer, and this is the biceps brachii, I'm only going to recruit the blue fibers. But if I was going to pick up, a milkshake or a cup of coffee, which is significantly heavier, then I'm going to recruit the blue and the red. Okay? So this is the concept of recruitment. Yes?
I was going to ask, does it work a lot, with your eyes, to see, how heavy you think an object is going to be?
Yeah, so you're going to have memory. You'll have memory over visualization of an object, and so it'll register in your brain that, that's probably going to be heavy. Right? And all of us have had the awkward situation where you see a backpack, right? And maybe it's full of, a jacket or workout clothes or something, no shoes. But it looks full and it's a large backpack, right? Can I borrow yours? Because this is a large backpack. Yeah, it is heavy, okay? So visually you can imagine this backpack fully filled, right? And you come and you think it's going to be certain weight, and you pick it up and you're like, oh man, it's, I was expecting it to be heavier. So there's an example of how your visual cues were telling you to recruit a large number of motor units, but it wasn't necessary for the task. Okay? Get you some extra credit for that. Nicely done. Just kidding. It's I just just playing with your emotions today, I know. So does that make sense, the recruitment concept?
So do the neurons recruit individual fibers or individual groups of fibers?
So the neuron itself is going to recruit individual groups of fibers based upon the number of fibers that it innervates. Okay? And we have different types of motor units. So that's where we're going next. So the idea here is it's dispersed throughout the muscle. So you kind of see that the reds and the blues, it's not like the left side of the muscle is all red and the blue side of the muscle is all, or the left side of the, right side of the muscle is all red, blue side of the muscle, left side of the muscle is all blue. Do you understand what I mean? It's not like they're segregated. They're actually intermingled so that you can still use the entire muscle, the entire cross-sectional area of the muscle, but you're only recruiting certain fibers within it. So it's not like the anterior side is one color and the posterior side is the other. You with me? So it's dispersed over the entire muscle. They will contract in unison when they're activated, because you want to have a coordinated contraction. They produce a contraction over a wide area. What does it mean by weak contraction? Well, an individual fiber relative to the entire muscle is not very strong. So some students get a little confused, like, what does it mean by a weak contraction? Well, because they're interspersed throughout, the amount of force that one fiber produces is relatively small in comparison to the entire population. But it's widespread because they're all doing it together. Right? This is kind of the analogy of a little bit from everybody in this room can go a long way. Right? That's the concept of like crowdfunding. We talked about crowdfunding with Runa Bega, right? That East Indian little girl that had this hydrocephalus disease. And with crowdfunding, everyone logged on, and, $15 here, and maybe $25 here, maybe $1,000 by one person, $2 by another. Pretty soon, they paid for her surgery. So this allows us to produce sustained contractions over a period of time, like postural muscles. So for example, postural muscles, you can probably tell if you work a long shift, whatever your work is right now, if you work a long shift, you can tell when your muscles are exhausted. And it's sort of, you're slouching a little bit more, right? You're not standing straight up. It feels good to sit down maybe. So you know that there's a limit to that, but you can still do that long shift, that 12-hour shift, that 14-hour shift. Okay? It gets more complicated as you get older, trust me. There's an effective contraction that requires the contraction of several motor units at one time. So most of our activities are going to need to recruit multiple motor units. But it's organized in this way where we can recruit systematically. Question?
So I just wanted to make sure, when you're working out, are you effectively just creating more skeletal muscle fibers, and that's why working out makes you stronger because you have more fibers to activate neurons?
No, usually when you're working out, what happens is these individual fibers, they get bigger.
Oh, okay.
So it's mostly, it's pretty much exclusively hypertrophy. So they enlarge versus hyperplasia, which would get, more of them. Which makes sense, because if you actually started replicating skeletal muscle fibers, you'd actually have to have another neuron come innervate it. But if you just enlarge the one that you already have innervated, it can do more work without trying to pull more circuitry. Okay? Question part B.
So then, but no matter how big the fiber gets, it's still going to be a relatively weak force?
Yeah. Relatively weak in comparison to the entire muscle, right? Yeah.
Does the body have preferences for motor units?
Yes, okay, let's perfect lead in. So if we look at different places in the body, on average, a motor unit, this is an average motor unit, okay? Humdrum, run-of-the-mill, average Joe, 200 muscle fibers for each motor unit. So one neuron goes to 200 muscle fibers. That's pretty standard. The question that you asked is, there are some cases, like when you look at the eye muscles or hand muscles, you actually want a fine degree of control so you have what we call smaller motor units, right? So you have 3 to 6 muscle fibers going or being served by a single neuron. So it's not a 1-to-1 ratio. It's a 1-to-3 or a 1-to-6. Does that make sense? So if I used an equation, you're going to want to pay attention to this, by the way. If I used an equation, I said, on the numerator I have the number of motor neurons, and on the denominator I have the number of muscle fibers. I could create a ratio of 1 to 3. Make sense? That 1 to 3 ratio mathematically is what number? You're like, oh crap. You can use your phone if you need to. 1 divided by 3 is 0.333 repeating, right? Those of you using your phone, confirm. Okay, thank you. Nicely done. If you look at the large motor unit, you're going to have 1 over, say, 1,000. Right? That's a large motor unit. It's 1 motor neuron supplying a large number of muscle fibers. That means that 1 signal generates a much larger force. It's not fine control, it's powerful contractions. And you would want that like in the leg muscles, like in the quadriceps, the hamstrings, or the gastrocs. You'd find that in the biceps, the arm muscles. You're not going to find it in the muscles that control the eyes or the hands. Here you want fine motor control, so you want more of a 1-to-1 ratio. So you want a larger number with respect to the ratio, equivalent to a smaller motor unit. Versus the smaller number, according to that ratio, 1 over 1,000 is a smaller number, but that is representative of a larger motor unit. Does that make sense, what we just walked through? Okay. So now to answer your question, I hope I've done it, is depending upon what strategy you need in different parts of the body, you're going to create either an average motor unit, or you're going to create a small motor unit or a large motor unit to give you this entire gamut of biomechanics that we actually appreciate and enjoy. Here and then here.
Can you give an example of average motor unit, like what muscle?
Somewhere in between. So a lot of the average one would probably be like rectus abdominis, muscles that are not huge, but they're not really, really small. Okay, like serratus anterior, sternocleidomastoids. You know, any of these, a lot of the supporting type of muscles that are synergists, but not necessarily the prime mover. The prime movers are going to be large motor units. Prime movers, like the main action. But our supporting muscles are going to have maybe more of an average number. Okay? All right. Oh, you had a question. Sorry, I almost blew you off, but I didn't. So the question is, if you have a motor unit, let's go back to this picture. If you have a motor unit that gets damaged or severed or has trauma, is there another motor unit that could take over for it? Yeah, and that's the whole purpose of not having the anterior aspect of the muscle only being served by these motor units and the posterior aspect of the muscle only being served by those motor units. I switched to anterior and posterior because I guess I get it better than left and right. You have redundancy, so you can recruit still the entire function of the muscle, but it may be compromised. You'll see that like in stroke patients. So if you see a stroke patient that's recovering and in PT, then their gait's going to be awkward at first, but as they train and recruit other motor units to do that same activity, they can compensate. They're not regenerating nerves. They're just recruiting new motor unit pathways that weren't necessarily leveraged before.
Does one motor unit control one muscle?
Does one motor unit control... A motor unit will only go to one muscle, but it won't be necessarily the only motor unit that controls that muscle.
Okay.
I don't know of an example in the body where you have one motor unit going to a muscle, and there's only one.
Okay, so my question from before was still kind of like, if you have several, like, will your brain want to say, I don't know, fire section 1 of motor units all the time, and if it needs more recruitment, then it goes to number 2, then number 3, but like 1 is the primary. That's what I meant by like,
Yes.
does your brain like prefer one? Like, I don't know, it's a terrible way to ask it, but I don't know.
No, I understand what you're saying. So the short answer is yes, and that comes with training. So when you learn, when you're training how to do a specific activity, you come in with a motor pattern, and you use that same motor pattern pretty much every time. Okay, and with training, you can hone that motor pattern. In fact, we've seen it in elite athletes. You analyze EMG traces and electrical activity of like pitchers, or a tennis player, or a basketball free throw shot, and those motor units that are being used are the same every single time. There's this one basketball player from U of A, last name Jefferson, Hollis Jefferson. And he's got this kind of funny little like shimmy when he shoots. Has anybody seen that? So he's up there and he kind of does this, and my kids are like, wait for it, and he's like, right, and then he like shoots his basket. But if you watch it, we've slowed it down on the screen. I was like, watch, I bet it's the exact same motion every time. Like, no way. I was like, yeah, and it is. He has the same exact little full body little shake every time. It's part of his free throw. In fact, the coach told him to stop doing it early in the season when he first came on. He started missing free throws because it threw him off. And they said, okay, fine. If you're going to make free throws, then do your little jig up there and let's move on. Okay, but it's his little signature. So, to answer your question, you and I have the same motor units that we typically use. Evidence of that is a stroke patient. If you or I had a stroke, and we survived it, and it was a motor efferent pathway that was affected, you would see it immediately. Our gait would be impacted instantaneously. It would take time for you to retrain area 2, area 3, area 4 to come on and do those activities. Okay? All right. The neuromuscular junction. So when we get to where these nerves now interface with the muscle, we've talked about the synapse. This is just another fancy way of talking about the synapse. It's called the neuromuscular junction. This is the point where the nerve fiber meets the target, and the target in our case in this unit is muscle. So the terminal branch of the nerve fiber forms this synapse with the muscle fiber, and a stimulation is able to occur at numerous points within this neuromuscular junction. So let's look at a picture. The picture shouldn't be too foreign. It should be very, very familiar. We have some new information down here that we haven't seen before, but everything up here, including all of this, should be reviewed. Another reason why I like to do nerve before I do muscle. But last time that we looked at the synapse, instead of having a muscle down here as our target organ, we actually had a nerve. So it was like a nerve sending a signal to another nerve. Now it's a nerve sending a signal to a muscle fiber. But acetylcholine is still the neurotransmitter. It's the neurotransmitter of choice for 201. It's the one that we elaborate on all the time. We have these membrane vesicles. They're filled with acetylcholine. They fuse with the membrane. They dump acetylcholine into the cleft. It crosses this cleft. You see these little blue rectangle boxes. Those are the acetylcholine receptors that it binds to on the other side. This acetylcholine receptor is going to open and allow for sodium to come in. If sodium rushes into this cell, which happens to be a muscle fiber or a muscle cell, what happens? An action potential, assuming that threshold is met, and that sends this electrical signal down the membrane surface. When it reaches a T-tubule, it dives deep into the muscle. Okay? Now, deep within the muscle is where we see the architecture that we've talked about, which is our striated pattern of thick and thin filaments as they overlap. You can see the multinucleated nature of the skeletal muscle. You can see the number of mitochondria because this is a highly energy-demanding tissue. Let me ask you this review question from this unit, though. The receptor here that acetylcholine binds to is what type of receptor? What type of receptor is it? Is it adrenergic? Is it cholinergic? Why is it cholinergic? Because it's binding acetylcholine, right? So it's a cholinergic type of receptor. If some of you said nicotinic, then you'd be also correct because nicotinic is a subclass of the cholinergic receptors, right? So that is the specific receptor type, is a nicotinic cholinergic type of receptor. Okay? And in this case, it's stimulatory because it's going to stimulate an action potential. All right, so if we look at the synaptic knob, right, that synaptic knob is the swollen end of the nerve fiber that contains those vesicles. That cleft is that gap between the nerve and the muscle is this cleft. The Schwann cell, you can see up here, the Schwann cell actually releases some of its membrane and allows the synapse to be contained. It isolates that neuromuscular junction from that surrounding tissue. So what that does is it prevents acetylcholine from quickly diffusing out of the neuromuscular junction because the membrane from that Schwann cell helps to kind of contain it within that space so that you can preserve the efficiency of making sure the signal is transmitted. If we look at what happens to acetylcholine, we talked about them being exocytosed. They go into the membrane. They bind to acetylcholine receptors. There's quite a few acetylcholine receptors in our body. And another feature that helps to ensure that the signal is preserved are these junctional folds. So the junctional folds are additional real estate, if you will, surface area, where you have receptors for acetylcholine so that you can maximize the opportunity of detecting the released acetylcholine. So instead of this just being one smooth surface, you have these folds that come in and you can add a ton more acetylcholine receptors. It increases the likelihood that you're going to detect that response. Now, if you lose receptors, and you don't have enough of them, you have muscle weakness. And that's what happens in myasthenia gravis. Myasthenia gravis is a muscle wasting or muscle weakness disease where you actually have an antibody that breaks down the acetylcholine receptor and blocks it. And so the electrical signal is sent, acetylcholine is sent out into the cleft, but it cannot bind to its receptor counterpart on the other side. So myasthenia gravis is a muscle weakness disease as a result of this receptor not being available to bind acetylcholine. Now our last structure that we need to kind of talk about is this basal lamina. The basal lamina is a thin layer of collagen and glycoproteins. Those are just proteins that are covered with sugar residues. And it separates the Schwann cell and the entire muscle from the surrounding tissues. Right? That's that membrane that I was talking about. So if you look at this basal lamina, I said the Schwann cell extends some additional membrane known as the basal lamina that contains or isolates and allows for that acetylcholine to stay there. Well, eventually you're going to get rid of acetylcholine because you don't want acetylcholine to forever be propagating an action potential in the synapse. So you'll release an enzyme known as acetylcholine esterase. We abbreviate ACHE. That breaks acetylcholine down into acetate and choline. Okay, those are its constituent parts. Those are reabsorbed. They're rebound and forming new neurotransmitter molecules to be used for the next cycle. Okay, so what does this look like? If we look at some other pictures, this is a high-powered light micrograph, histology, and you can see these muscle fibers. You can see the motor nerve fiber coming down, and you can see the neuromuscular junction that broadens out right here as it innervates that particular muscle fiber. If we look at some other images, here's a high mag view. Again, we're going to pseudocolor some of these. So here's our skeletal muscle fiber in this pseudocolored blue. Here's axons of motor nerves coming down. You can see this one goes right here. This one goes right past it, and obviously synapses down here. This one dives deep into the screen, three-dimensionally, into the muscle fiber behind this particular fiber in our plane of view. And then this is a new term for us. This is the motor end plate. The motor end plate is another term for the other side of the synapse. The motor end plate is another term for the postsynaptic terminal when it's a muscle. When we talked about nerves, we said we had a presynaptic terminal, we had a postsynaptic terminal. When we talk about nerves innervating a muscle, our postsynaptic terminal is referred to as the motor end plate. Why the motor end plate? An end plate is an electrical terminology that says this is where the electrical signal is supposed to stop. This was the destination. It's called a motor end plate because it moves, right? It's a muscle that actually can do an activity. So I don't want you to be confused if you see that term on the exam. We talked about it, and this is our analogy to our postsynaptic terminal. We look at some other pictures. Now we're moving out of histology. We're going into electron microscopy. This is our neuromuscular junction under high magnification transmission electron microscopy. Okay, instead of using light to bombard the sample, we use electrons. And you can see that this is our primary synaptic cleft. That's the gap. If you take the gap and all of the folds that increase our surface area, the secondary synaptic cleft are these junctional folds. So if you took that surface area and you stretched it out, you would take all of the primary and you would add in the length of this, and you would get this very large surface area that's full of receptors that enables acetylcholine to bind. Up here, you can see sort of the punctate dots of blue. These are the synaptic vesicles that are on the presynapse side, containing the acetylcholine before they're dumping into the cleft. And then here you can see the mitochondria that are found in that synaptic terminal. It's rich with mitochondria because there's a lot of activity that's happening here. You're going to see shortly in the lectures of today and Monday that we're going to come up on the ATP demands, the energy demands, adenosine triphosphate, and that's mostly manufactured by these mitochondria. If we continue with this electron microscopy viewing, you can see the skeletal muscle fiber is shown here on the right, pseudo-blue. You can appreciate the primary cleft right here. And these little folds or invaginations are the secondary cleft that again is increasing the surface area, allowing for more acetylcholine receptors to be inserted into that membrane to encourage a more robust response. Again, primary cleft, secondary cleft, or junctional folds is another terminology for it. Here you can see the mitochondria, and here you can see the synaptic vesicles. So a number of different views highlighting these different features that we're looking at this NMJ, Neuromuscular Junction. It's the junction between the nerve and the muscle. That's all that word means. Okay? Questions?
Will images like these pop possibly be on the exam?
Absolutely. Reasonable question. And yes is the answer. Could you see pictures like this? Yeah, absolutely. Okay. I would understand what you're looking at and what's happening. Okay, they're really, I picked a pretty blue, so you can, like a azure blue or something, isn't it? Maybe it's, is it teal? I don't know. I don't know. I like it. It's nice. It's calming. It'd be a calming test question because you'd be like, oh, it's a nice calming blue. If it was like red, you'd be like, oh, right? All right. Neuromuscular toxins. Okay, so why do we flip and care, right? Sweet, You got blue pictures, stuff's happening. You got these folds, we're releasing these neurotransmitters. Okay, great. So why do we care? Well, here's why we care. There are some clinical ramifications to what happens at these junctions. So let's look at some toxins that interfere with the synaptic function. There are certain pesticides, actually, and they contain these cholinesterase inhibitors. So that's going to bind to acetylcholine esterase and prevent it from breaking down or degrading acetylcholine. So now if acetylcholine doesn't go away, what's the result? If acetylcholine stays perpetually in the cleft, what's it doing? Stimulating contractions. Okay? So now you have what we call spastic paralysis. You're paralyzed because you can't stop it, but it's spastic because you're in a continual state of muscle contraction. Okay? The problem with that continual state of muscle contraction is your respiratory muscles have to contract and relax to actually move air. If you just contract, you only move air in one direction. Right? Everyone take a deep breath and then just hold it for the remainder of the lecture. Okay? You'll pass out eventually. Okay? If you exhale and hold it, same result. Okay? Tetanus. This is a form of spastic paralysis caused by a toxin known as Clostridium tetani. So in this situation, the toxins block glycine release, and that normally glycine stops motor neurons from producing these unwanted contractions. So instead of like kind of having sort of a shaky pattern as you're pointing to the bathroom, right, you have a nice smooth pattern because of the effects of glycine. Okay, so this tetanus toxin blocks glycine release. You overstimulate and you spastically paralyze the muscles. And if you do some Google searching on lockjaw images from old, vintage or historic lockjaw images, you'll see patients that are like this. Okay? So they're contracting these muscles. They're usually sitting on a table, like laying on a table, right? And their jaw is like completely tense. Okay? And so this is spastic paralysis. Eventually, if they're not treated and you can't get to them well enough, then they would suffocate. Okay? So they can't pass air. The spastic paralysis, flaccid paralysis, is just the opposite. So this is a state where the muscles become limp. They cannot contract. They're in a perpetual relaxed state. Curare is a drug, naturally occurring drug. It's a poison. That competes with acetylcholine, so it competitively competes. It binds preferentially to acetylcholine receptors and prevents acetylcholine from binding. And so if acetylcholine can't bind, you don't send the action potential to the other side. The motor end plate never gets the signal. Curare is a plant poison that's harvested. It can be found in South America. And this is the active agent that's used by tribes in that local area to tip the darts. It's poison that goes on the blow darts. And that's how they paralyze their prey when they're hunting, or even, other competitors that are in the area. Another example is botulism. So the neuromuscular toxin that's from the bacterium Clostridium botulinum. You guys remember where we saw that before? Well, where did we see that before? Botox. So that's what we talked about previously. but you can find this, unfortunately, in foods. Okay, and this obviously blocks the release of acetylcholine, like what Botox does with Botox injections. And that causes flaccid paralysis. So the flaccid paralysis is the muscles don't contract. If you're injecting them into the face, then you get relaxation of those muscles and the wrinkles disappear. Botox is also being used now. I think it's still considered off-label, but it's probably going to be soon on-label, meaning the agency will approve it. But it's being used to treat migraines. And part of the reason for that is one of the thought processes is that there's multiple things that could cause migraines. It's not just muscle tension. But if it is muscle tension related, by paralyzing the muscles, okay, maybe in the back of the neck, on the top of the head, to kind of relax what's going on. You know, unfortunately, it may not be just the ones right here, right? It might be ones that are located around the head that are causing stress onto the head and causing a headache. Okay? But one of the more commonly used features is actually for wrinkle removal. I want to introduce this concept, and then we're going to pause and I'll let you have time to pick up your thing. I want you to read ahead for Monday, because this is really, really important material. This is the mechanism of how it works. Okay, so I want you to look at the remaining slides in this particular lecture. There's about 9 slides of content. And we're going to, actually there's not even 9 slides. There's actually about 6 slides of content, and there's a video. It'd be super advantageous for you to watch the video before Monday. Okay? We're going to pick it up with the slide of filament theory, and as you're packing up, just try to be a little quiet, because I want to explain this. We get these filaments that slide across each other. Okay? And we're going to unpack in a little bit more detail how they slide, what causes them to slide. If you're leaving and you're leaving your notes, now's the time to pick them up. They're there in the back, alphabetized by your last name. If you turned it in, you didn't get credit last time, and you want those to study, now's the time to get them, because you won't see me again until Monday. So if we look at the sliding filament theory, and we pay attention to what's going on, we can see 3 different states. Okay, we can see, I'm going to keep talking because we still have 3 minutes of lecture. You guys can decide if you're done or not. But some of the most difficult questions on the exam are going to be on this. Just FYI. So here's a relaxed muscle. Here's our sarcomere. And you can see in a relaxed state, the Z discs are pretty far apart. Z lines. You can see as it contracts, the Z lines get closer together. As it's fully contracted, the Z lines are in the closest configuration. These are the drawings or the schematics. These are electron micrographs of what that actually looks like. So you can appreciate here very visibly is the Z line or Z disc. And you can see in the contracted state that you have a lot of overlap of the thick and the thin filament. Okay? The amount of force production that you're generating in these different places is actually directly related to the number of interactions between actin and myosin. Okay? And we're going to call and talk about that on Monday. They're called cross bridges, because they actually form a connection and then they move. In the video that I want you to look at, this is the last slide, this won't pick it up, but the video I want you to look at is a YouTube video. We'll come back to all these, don't worry. And this is the link. And if you can have the visualization of how it works for when we start on Monday, you're going to be ahead of the curve. Okay? Have a nice, wonderful weekend. Don't forget to pick up your notes in the back. I promise you can get them back. Now's the time to grab them. There's still about 2 minutes left in class.
Yeah.